Yeah. So actually, my thought was just as a working session here to try to get the frame. We go relatively quickly through the biochem. Feel free to ask questions. I'll go as deep as you guys want to go,
Yep.
and then kind of build up to the current drawing that I have. So, starting briefly, y'all are familiar with the notion of ATP, right?
Indeed.
Let me share my screen here. Share screen, window, this one, share. Can y'all see that?
Mine's loading still.
Same here. It's on its way. Oscillating purple dots.
In theory.
Come on, Riverside.
There we go. All right, so y'all can see there the ATP, ADP. By cleaving off one of the phosphates, it releases available energy. So ATP functionally becomes the sort of energy currency unit of the cell. Production of ATP is the functional end result of metabolism, right? For our intents and purposes. That cleaving of ATP goes to muscle contraction, et cetera, can be used in the cell in various ways. So how is that done? So glucose here comes into the cell across one of these channels.
Quick aside, if you
Yep.
can zoom a bit, maybe for whoever else may be watching this as well. Yeah,
totally.
Cool.
Yep. Comes in, is broken down progressively into pyruvate or lactate. New evidence indicates these two should be reversed now, but minor biochemical quibble. This is how it was taught. It releases 2 ATP, right? A very small number of ATP, but it does release some. NAD or NADH is converted to NAD, and when NAD is converted back to NADH in the process of this reaction, that's how the ATP is released. Only mention that because now people are trying to promote like IV infusions of NAD, with the idea that it'll boost this. It won't, because this is basically already at maximum. This is a very tiny number, and all of the NAD inside the mitochondria cannot be crossed. So NAD does not ever cross the mitochondria, right? So no matter how much you supplement in the vein, you're not going to change anything in the mitochondria. Anyway, pyruvate comes down, is converted into acetyl-CoA, and I'll get to that in a second. In the process of that conversion, again, an NAD is made into NADH, and a carbon dioxide is released. So pyruvate is a 3-carbon molecule. Acetyl-CoA is a 2-carbon molecule. And
actually, and if you don't mind, and it may pain you, and if it's too early to stop, that's okay. But if we go back to that picture, I was just curious structurally about the primary boundaries we're seeing. I see blood and systole divided by a boundary at the top, which is demarked at the far right by PM. Is that, what is this boundary? And then there's another boundary, of course, that is similar thickness, but orange. And that might have been the mitochondrion, as you were discussing.
Correct.
As an organelle? Okay.
Yeah, so, no, great distinction, actually. So, blood and cytosol, as you pointed out. So at the top is blood. This brownish beige area is the inside of the cell.
Okay.
The orange membrane is the membrane. The outer membrane is mitochondrial outer membrane, MTOM, right? Mm-hmm. And the mitochondrial inner membrane, there is actually a matrix inside the mitochondrion. So there are two membranes there actually within the mitochondria. So
got it.
You know how that was discovered? Is this electron microscope in it still?
I don't know if that's how it was originally discovered. I know you can see it under an electron microscope, but I don't know the history of it.
Okay. Thanks for that for the biologically sort of naive here.
No, that's great. Yeah. No, it's an important, and it'll become important later on. So I'm glad you stopped me. So the TCA cycle here that you can see, right? We're going to zoom in on that. Here is acetyl-CoA coming in. It's a 2-carbon molecule. Each one of those is a carbon. The cycle loops back on itself. So you start sort of with a 4-carbon molecule, joins with 2, you end up with citrate, which is a 6-carbon molecule. Citrate, you peel off 1 carbon, you release carbon dioxide, you convert an NAD to NADH. Now you have 5 carbons. You repeat that, NAD to NADH. You keep going. FAD and GTP. GTP is just sort of another, it's an equivalent molecule to ATP, as it says. FAD is similar to NAD, just slightly different nucleosides involved. Another conversion here, NAD to NADH. So the final, and then you end up back again with the 4-carbon molecule. So basically, you peel off 2 carbons as carbon dioxide, and you end up with functionally 4 conversions from NAD to NADH, right? And 1 ATP, right? So
the oxygen is available already in the cell, or it's part of the molecule?
yeah, so it's part of the molecule, actually. Let me show you here. Nope. There, you can kind of see it better here. So citrate is carbons with oxygens, right? So when it peels off one of the carbons, right? So actually, so this carbon gets peeled off. It already has 2 oxygens attached to it.
Okay.
Anyway, that's the TCA cycle. Point is, it produces a little bit of ATP, a little bit of carbon dioxide, and a whole bunch of NADH, right? The NADH then goes to, this is electron transport chain, ETC. It goes over here, and we'll zoom in on that. And this whole process happens between the 2 membranes of the mitochondria. So, there we go. We can zoom in there a little bit. So NADH is converted back to NAD, and hydrogen is pumped across this across this membrane, right? So 4 hydrogens are pumped across. It's in that conversion, it donates 2 electrons. The electrons travel down this chain, a series of reactions, and at several stages, the movement of the electron allows for transport of hydrogen over this membrane, right? So through complex 3, as the electron passes through, it pumps 4 hydrogens, comes through to complex 2. Then as it goes across complex 4, 2 more hydrogen pumped across. At the end of this whole electron transport chain, the electrons are dumped into joining an oxygen molecule and 2 hydrogens, and produces water from oxygen. That is that's the whole reason we need to inspire oxygen. So all the oxygen we breathe serves as the final receptor of those electrons to be converted into water. So the whole process
this is still mitochondrial. This is in the...
Correct. Still in the mitochondrion, in between. So the hydrogens are pumped into this space between the 2 membranes.
Okay.
Right? So what ends up happening is, in this in-between space, you end up with a very high electrochemical gradient, right? Because all the hydrogens have been pumped, so that's highly positively charged. The inside of the mitochondria is highly negative charged. And by allowing hydrogens to pass down their electrochemical gradient, ADP is converted into ATP by this enzyme. After it's been converted, it uses this transporter to exchange an ATP out and get an ADP in to the mitochondria. So this transporter uses that same electrochemical gradient, but it uses it to communicate. It bridges actually both membranes and exchanges an ATP from the inside of the mitochondrion for an ADP from the cytosol. With me so far?
Yep.
But you're
So
saying the electric charge difference is what causes that to happen.
Yeah, correct. That's also provides a pumping. It's actually almost a physical pump. It literally, it literally like has this conformation, right? Binds to an ADP. The binding forces it into this conformation. It releases it. The ATP floats here, and then the electrochemical gradient allows it to release back. So it's literally pumping an ADP in and an ATP out with every cycle change in a 1-to-1 ratio.
and that's happening microscopically across the membrane, that surface then essentially.
Correct. And then you have free ATP in the cell to be used for whatever cellular actions are required, right?
Mm-hmm.
Briefly mentioned this creatine. So creatine, ultimately, the best way to think of it is through a series of reactions of exchanging phosphates, creatine can gain a phosphate that can be donated freely to ADP to convert it back to ATP. So it almost serves as like a phosphate sink, if that makes sense, to allow the ADP to be turned into ATP from phosphates that are just sort of stored on creatine molecules, right?
Got
So
it. So
that
the
little-
ADP is available, and the presence of creatine and
Creatine
phosphates
phosphate.
then.
Yep.
Gotcha. Yeah, it is what enables that little extra boost in ATP.
So you get ATP a little bit from glycolysis, ATP a little bit from the TCA cycle here, right? And then this product produces a ton of ATP, right? Because it's just cycling it down its gradient, right? And so you get 2 here and 2 here and like 30 here, right? I don't know if that's exactly right, but it's on that order, if that makes sense. So that I think is enough to work with. So this is my schematic of the first process, glycolysis. Oh, sorry, no, I'm missing one. So this is glycolysis from glucose down through this cycle. You can also, from free fatty acids come in, they permeate all the way into the mitochondrion, and through beta oxidation, match up at acetyl-CoA, and it's the same from that point forward.
Are those sort of superimposed? Like, are those processes additive in a sense? Like, if one or if both are performing, does that end up being a bottleneck where they merge in that sort of visualization, or does it end up being kind of supplementary that you'll get more throughput if both are active?
Neither of those, I think.
Got it. So there's no limitation at the convergence of those vectors. We could look at the diagram again if that's helpful. But also,
yeah. So what's interesting is that each of these molecules kind of serves as and that's what I was about to show. The steps are regulated, right? Because each of these events happens by an enzyme that acts as a catalyst. That enzyme is subject to inputs about how well or badly, or how fast that it is able to accelerate that reaction. So a buildup of acetyl-CoA acts as a negative inhibition to the production of pyruvate and glycolysis, right?
I see.
So it creates back pressure if it's being supplied through the fatty acid chain.
Got it.
Right. So let me see if I can subtract some things here. Okay. Starting with, so this is glycolysis, right? Glucose, G6P, F6P. We had to memorize all these in med school for no fucking reason.
Have you ever pulled it out?
The actual names are so irrelevant, it pains me that they still teach them. Like, you need to know that like, the steps are important, but like, the names are not. But whatever, it's a way of testing, I guess.
Those that can't.
I guess, yeah. Anyway, so production all the way down to pyruvate. Again, new evidence that it actually always produces lactate. So lactate should probably be considered the final product, but it's been taught as pyruvate for a long time. That'll probably have to be revised now. Anyway, and again, produces a little bit of ATP in the process of producing that pyruvate. The important bit is that each step is regulated by enzymes, right? Which are subject to back control. So glucose convert to G6P. If G6P piles up, it negatively inhibits the enzyme that produces that reaction, right? F6P.
Actually, I have a little
Yeah,
bit of a gap. Can we talk just enzyme in general?
Sure.
I know, talked about the generalities, but it's which part of the cell is it intercellular inside the cell mechanism?
Yes.
But isn't it also something that's happening in between cells?
Well, yes. Enzyme
Like
is
when you
a
say
general
metabolism-
term for reaction catalyst. So an enzyme is a protein that has sort of physical and electrochemical conformation that makes it fit really well with certain molecules, right? So like this enzyme will bind to glucose quite well in such a way that it brings the desired reaction into a higher probability, if that makes sense. I don't know how well I explained that.
Well, no, yeah. I guess, yeah, but when you, like you mentioned back pressure, like there's got to be like information theoretically. Is it the ionization that's causing the reconfiguration of the enzymes due to the back pressure?
It's actually a lot of things. So yeah, enzymes can be regulated by things like pH, can be regulated by electrical gradients, but can also be regulated by partial binding. So G6P may partially, and I don't actually know if this is how it regulates it, but just as an example, it may partially bind this enzyme, not as strongly as glucose, but enough to interfere. If there are a ton more of this than this, if there's a ton more G6P than glucose, then you may end up with glucose competitively unable to access the enzyme and thus the reaction not be able to happen.
Okay, so there's a bunch of states, all these different dimensions like acid to base and electrical. They have the intelligence of what to do is a combination of those states, and there's different balances are hit.
Yeah, I think the
Yes,
primary, and actually even with pH and stuff, ultimately pH regulates the enzyme by affecting binding affinity. So the final arbiter is how able the enzyme is to bind to its desired substrate.
Okay, cool. I mean, I've got a general picture.
Sure. Yep. Okay, so point is, if G6P builds up, it inhibits negatively. If F6P builds up, it doesn't inhibit the immediate enzyme. It actually ends up inhibiting the same enzyme, right? What's interesting is that if
does red generally connote inhibition
correct.
in this diagram? Is that something? Okay, so
yep, green. I've used green for positive facilitation. So what's interesting is that if you cross this threshold and you get to fructose 1,6-bisphosphate, it actually facilitates this enzyme and increases its affinity. So it speeds up the reaction of pulling it to pyruvate. So if this builds up, it actually produces a cascade, right? So there's kind of almost this cut point here where if the reaction has proceeded to this point and there's a lot of this, the way the stimulation systems are set up, it's designed to push that forward very rapidly. Down this, each one of these is a step that I didn't bother to list, up to pyruvate, right? Similarly, if there's a bunch of ADP, right, meaning ATP has been used up, that facilitates this enzyme's action from F6P to, right? AMP also stimulates that quite strongly. By contrast, if there's a bunch of lactate buildup, that inhibits this enzyme, right? And if there's a bunch of ATP, that also inhibits this enzyme. So this conversion from fructose 6-phosphate to fructose 1,6-bisphosphate is one of the key kind of control areas where there's a bunch of different inputs, some of which enhance the function of the enzyme, some of which degrade the function of the enzyme, controlling kind of communication, if you will, about the energy state of the cell.
Mm-hmm.
Right? So then you enter the citric acid cycle. I mentioned acetyl-CoA kind of goes back and inhibits this conversion, right? Citrate also is a negative control all the way back here. Now, some of this is less effective because you've crossed the mitochondrial barrier here, right? So there has to be quite a bit of pileup before that's communicated back, if that makes sense. You go through the cycle, you get a bunch of NAD, right? Which then produces ATP. And this is just further communication of sort of like the full spectrum of control inputs, right? Like a lot of things have positive and negative stimulus through this process.
And all of the green and red lines here, they connote the influence on some enzymatic behavior. This is all influencing catalysts. In other words, there's an ongoing reaction and there's a regulatory effect by these enzymes, and these things are all influencing the amount of or the performance of in some way of those enzymes.
Yep, exactly.
Got it.
Right. Mm-hmm. So schematically, this is kind of where we end up, right? And so what I'm trying to represent here is pools of energy substrate. So here, liver glycogen and fat, these are tissues, right? From tissue, this is the bloodstream, this middle column here with blood glucose and free fatty acids, right? This is the cytosol, right? This is the cell, and this is the mitochondria in this far right lane. And the way that as you can see, this is usually taught because it kind of makes logical sense that you move from glucose to ATP, or you move from fatty acids to ATP. But in fact, part of the issue with that teaching is that and this was the hardest thing for me, maybe other people got this more readily, but it puts you in the cycle of thinking of it as a push process, when in fact it's a pull process, because depletion of ATP is the thing that starts speeding up the whole reckoning. So ATP is a negative inhibitor of this, conversion from glucose to pyruvate, and the removal of that inhibition is what starts speeding up the cycle, if that makes sense.
It's a sort of, maybe in some ways, and I may be oversimplified, but it's a classic kind of closed system set of semantics here, where it's like, well, moving things on your right hand is going to influence things on your left, and vice versa. And so depending upon which part of this whole cycle or set of reactions you're looking at, you can think of it as kind of push or pull, but ultimately those two things are tied together.
I mean, maybe we can talk this out, actually. I think that the issue with even conceiving of the notion of a push is that the stimulus always comes from downstream, right? Because as you do
or from
activity...
upstream? Just trying to make sure
from ATP, or the depletion of ATP.
Okay. Oh, I see. Okay, I'm with you. Yeah.
Right. So as you burn ATP, it gets depleted, and that is right. There's no, but there's not a primary way for your blood glucose being high or your cell glucose being high to make this reaction happen much faster. It would happen a tiny bit faster, but you're not playing on any of the control mechanisms, right? The control mechanisms.
Bandwidth on the wire.
Correct. But the depletion of ATP increases the bandwidth by removing the negative control. if that makes sense.
Reduces some resistance.
I see. Is the cellular effect of like pushing glucose or something? Were you getting at there that inevitably that might be the energy source, or in some way chemically important to these other things all happening, but it's kind of only in a supplementary way? Ultimately, if we view this as a system, the primary components, in this case, ATP depletion, is the primary mechanism by which you can sort of start pushing the pump again.
Yeah, ATP depletion is the primary way, right? If you build up like cell glucose, it's more likely for that glucose to be pumped back out of the cell and made into fat or made into glycogen or like something else. Like, because this pathway, if ATP is pretty well repleted, is just not going to be open functionally.
Gotcha.
So increasing this is not going to push the process forward, whereas depleting ATP will very quickly start pulling the process forward, if that makes sense.
Mhm.
So to me, the analogy that came to mind here is the notion of a tank with a faucet at the bottom, right? So, and almost like a toilet, right? When the water level drops below a certain point, it opens a valve that starts flow down from a particular set of pipes, right? If the water level drops even lower, it triggers another valve that opens another set of pipes, right? With the intention that the tank should never empty. That's your body's goal. Now, the faster you're burning energy, the more wide open that faucet is at the bottom, the greater the rate of drop in that tank, and the more valves will start opening, right? And water will start flowing down them, if that makes sense.
Mm-hmm.
So the first valve, if you will, is from fatty acids. So fatty acids from the bloodstream go all the way into the mitochondrion and join at acetyl-CoA, just as I showed, goes through the TCA cycle, produces all that NAD, and ultimately produces ATP. So this cycle here is the first valve that's turned on in a healthy human, right? The width of the arrow here is intended to convey speed of reaction, right? So the transport into the mitochondrion is relatively slow, and so despite other reactions being relatively fast, there's a bottleneck here with the transport from the blood into the mitochondrion. So the ability of this to you can only run the faucet at equilibrium so fast on this basis, right? If you just you're just walking, well, you're not burning enough energy to overwhelm this pipe system. And so that valve will be the primary valve that you use, right? Other...
This is kind of like with metabolism. We were just talking like the first minute or whatever. Of like,
What's that?
Like the first minute, if we're thinking like a human body and exercise, like we're pulling. Crap, I'm forgetting all the stuff for.
No. Yeah. So this is the
Is it fatty acids that you're pulling before you start doing like the oxygen?
Well, so this uses oxygen, but yes, yeah. It's a more efficient reaction as well, right? So it produces. Let me see if I can get this right. Actually, let's double check this. So RER ratio fat. Right. So so the respiratory quotient is something that we use. It's the ratio of carbon dioxide to oxygen use, right? And so, the ratio of carbon dioxide production to oxygen use. And so fatty acid burn only produces 0.7 carbon dioxides for every oxygen that's used, whereas carbohydrate burn produces 1-to-1. And part of that is just you're entering the cycle here rather than this additional step which produces carbon dioxide, right? So the conversion of pyruvate to acetyl-CoA produces a carbon dioxide, whereas fatty acid to acetyl-CoA does not. And so you get a little extra carbon dioxide, bringing up the ratio to 1 from glucose metabolism. Anyway, point is, this is an efficient system in terms of like producing less carbon dioxide for oxygen used. You also, and this is not to scale. I tried to make things to scale, but if this were actually to scale, it would dwarf the entire graph, right? So the other thing I was trying to represent somewhat unsuccessfully here is sort of the size of the pool, right? So ATP is your body doesn't keep a bunch of ATP floating. It keeps a little, but not a lot. Creatine, it keeps a little more, serving as kind of a pool for the ATP, or a pool of phosphates to immediately regenerate ATP that's burned. But the pools are relatively small. So when you open the faucet, immediately the ATP sort of like is depleted. You burn a little creatine to replenish that, but the ADP that's produced sort of turns on this fatty acid, it's never off, right? That's the other thing I guess I should say is, if it were off, you'd be dead, right? So it's always trickling out. So we're just, you never get to the fully off position. But if you increase the rate of flow, the flux initially is from fatty acid, acetyl-CoA to ATP. So like I said, the intention to some degree was to say, okay, size of pool in terms of how much energy it can produce. So how much ATP can this produce? And then the size of the arrow here to some degree is the speed of reaction. But this pool actually would be incredibly huge if it were to scale, right? Like the amount of, even if you're super lean, like the amount of available energy stored in your adipose tissue is insanely huge. The issue is just the speed of reaction and transport, right? So you open the faucet a little bit wider, right? As you open the faucet a little bit wider, this gets even more depleted, right? And starts turning on your glucose burning system. So now this acetyl-CoA tank also has dropped, right? And so it also turns off its negative inhibition here. I can't remember if no, I didn't create that. Whoop.
Are you guys hearing a slight buzz?
Yeah.
It's okay. Bees make honey. We can deal.
Yeah, that's Danny's. I don't know. What's buzzing? Anyway. Oh. that was a nuclear solution to the problem.
Mm-hmm.
Anyway, we'll wait till he gets back. But
no worries. Okay, so even zooming out before he gets back, we can,
if
this is relevant, we can fill him in. But so the goal at some level is, or one of the goals, I suppose, and thanks for all the education along the way, is to try to find a perhaps better or good, at least, representation of the key components to these metabolic processes, and in doing so, then better communicate sort of some of the punchier potential interventions and things of that nature to patients and others, into the world. Is that kind of
that's exactly
that's
right.
the goal?
That's the goal. Yeah. And we've already kind of hit the key issue, so I can even kind of speed up here. But the key thing that I'm trying to say is that number one, the system is driven by the rate of depletion at the final step, right? If you're walking versus sprinting, this pool is being depleted at a different rate, right? And the second bit of information is that depletion triggers flow by removing inhibition, right? A valve is opened that was not open, or opened wider that was not open.
Mm-hmm. And that mental model that you were using, actually, one of the visualizations that I had in my head while you were explaining that was something to the following effect. And if this is not right, then that would be clarifying as well. I'm imagining, some cylindrical container vessel, and there's water in it. And as the water level drops, right, it's almost as if there are sensors with corresponding, I think you call them valves, sensors and corresponding holes in the container. And as the water level drops below sort of that, that level, water will suddenly be turned on to begin attempting to replenish or refill the tank in that regard.
Exactly right.
Okay. And so there might be at some level, that analogy kind of holds up a bit, because what you get is the ability to maybe, modulate the position vertically in the tank as sort of a measure of like how important things become at various levels of depletion.
That's exactly.
But then also you can have things be at similar levels in the tank, just sort of radially, if they kind of engage at the same time. And then the size of those holes might be indicative of sort of the amount of impact they have on ultimate ATP. Okay. And then there is, as things fill back up or so, that's the back pressure we've been discussing, rather, where ATP is, as there is if it's available in abundance, then you get this back pressure enzymatically that prevents, the cell glucose and other things from, Exactly. And is the back pressure, that back pressure is that a unifying concept in a sense that all of the pathways that produce ATP will ultimately be back regulated by an abundance of ATP itself?
Yes.
Okay.
Yep.
Okay. Yeah, that's helpful for modeling in general, right? That there is a principle on which we can lean pretty heavily for whatever abstraction ends up kind of being able to articulate this conveniently and comfortably for those who may not dive into this level of detail, which I realize is still a very kind of cursory level of detail even. So cool.
Yeah, no, I think, yes, I think you've articulated the core concept exactly. So anyway, adding just a little bit. So speed of reaction starts to matter exactly in the way that you described, because let's say taking this acetyl-CoA, right? The water levels dropping, or the level of acetyl-CoA is dropping. It drops, and so your body maximizes fatty acid production, turns on epinephrine to burn more fat, to release more fatty acids into the system. It's transporting across, but there's only so fast this can happen, right? So if the level falls below a certain point, then your body actually turns on the glucose pathway, because those pipes are bigger, right? The speed of these reactions is just way, way faster. However, the pool size is much smaller. So the available total, cell glucose and muscle glycogen, these are faster reactions, but there is a limited supply compared to the amount of energy from fat, right?
Mm-hmm.
So if you are running, at a moderate pace, right? You'll be burning, let's say, a mix of glucose and fatty acid, right? Both systems are turned on. And because there is free flow, you're not building up a whole lot of hydrogen. I didn't emphasize that very much in my diagram here, but in each of these steps of the TCA cycle, it's not shown actually very well there. Here, in this diagram, NAD is produced, carbon dioxide is produced. It's NADH, and a free hydrogen is produced, right? So that acidity is actually one of the things that sort of like limits this whole system. It's actually one of the things they think, along with inorganic phosphate, that creates muscle soreness, is the acidic damage, right? Anyway, point is, if there's free flow, you are producing some acidity from this process, but you're able to buffer that with your bicarbonate and carbon dioxide system, and you're basically able to regulate by breathing out enough functionally, and I know people who nitpick will nitpick this, but functionally carbon dioxide acts as an acid in the blood, right? And so as you exhale carbon dioxide, you alkalinize your blood, which is why your breathing speeds up as you work, because you're producing this hydrogen, which then increases, and carbon dioxide as well. So you're producing carbon dioxide, you are also producing hydrogen that converts bicarbonate into carbon dioxide, which you then exhale as a means of balancing the pH of your blood, and by default as well, the pH of your cell, right? So rate of production of hydrogen and rate of production of carbon dioxide are regulated by your exhalation system.
What happens? How is that hydrogen bound when we exhale the carbon dioxide? Because we're not losing, it looks like, a hydrogen there, but it's
No.
not free, and so it doesn't have the same acidic effect, which sounds like it has downstream implications for
Yeah, it's a
how you
bicarbonate.
regulate.
Yeah, it's called the bicarbonate buffering system.
If it's too much, it's fine. It was more just a curiosity. I don't know if it has any implications at all.
I mean, it's pretty quick. We can quickly, here, this carbonic anhydrase. So free hydrogen with bicarbonate forms carbonic acid.
Oh, it's water. Okay, there you go.
And then water and carbon dioxide. Yep.
Yeah. All right.
So, anyway, point is, at some point again, you overwhelm your body's ability to maintain pH, right? If hydrogen is being produced fast enough and carbon dioxide is being produced fast enough, then at some point the acidity builds up, right? Which is a limitation the fatty acid system does not have, right? Because it can never happen fast enough to where you're not able to breathe off that carbon dioxide. Whereas glucose metabolism can happen at a rate that produces hydrogen and carbon dioxide at a rate that exceeds your ability to compensate. Anyway.
Okay, somewhat intervention-y question. Yeah, we're taking you on multiple tangents. It's not great for focus. My apologies. So the thinner blue line, this fat to fatty acid pathway, that thin blue line, are there known mechanisms to, in the metaphor here, in the analogy, grow the thickness of that line, increase the sort of amount of that reaction that can occur?
Yes, actually, yeah, great question. So zone 2 training does that, right?
Okay.
And it's interesting because the way it does that, it actually doesn't affect the reaction speed. It actually increases the total number of mitochondria, right? So you just have a greater number of mitochondria available to be doing this concurrently.
Mm-hmm. You get more reaction. Makes sense.
Yep.
Yep. Very cool.
I mean, as with anything in the body, right? There are theoretical maximums. Like, very, very highly trained athletes can burn fatty acids at a rate of 12 kilocalories per minute or whatever, right? So like their maximum fat oxidation can be quite remarkable. Whereas for a general athlete, male athlete, anything above 8 is actually really quite good. Anything above 6 is decent even, right? But as you move towards unhealthy, like diabetics and so on and so forth, that can drop really, really low. You can get people that can only burn 2, kilocalories per minute, right?
Mm-hmm.
And one of the ways you measure that is your lactate, right? So if this is the primary pathway, you're not going to have a whole lot of lactate building up because lactate is generated by this pathway. And so you can measure the lactate levels in the blood. That's the notion of the lactic threshold, right? Is, lactate will build up, and then it'll build up exponentially as you overwhelm so it builds up linearly, and then as you overwhelm your compensatory mechanisms, it builds up exponentially. And that transition from linear to exponential is the lactate threshold.
Interesting. Do we know why zone 2 in particular ends up fattening that thin blue line?
So I do not know molecularly, but on the basis of principle, just like anything in the body, if you use it more, your body facilitates using it more. So if you start working at a level that only allows fatty acid, right? Then just like strengthening a muscle, your body will compensate by upregulating that process.
Mm-hmm.
So I don't know the
but it's
sort
more
of molecular.
You're saying more mitochondrial glycogen. Those arms. Is it ribosome? It is?
No. I mean, just more mitochondria. Probably the best way to say it. More mitochondrial density, right? and total fat oxidation. So all the enzymes are present in higher numbers because you have more mitochondria, et cetera, et cetera.
Actually,
I suspect the efficiency of each mitochondria also goes up, though I don't actually know that. But
so my very weak and sad. So this actually is making me think that should have prepared for this discussion and actually could have been more insightful along the way or asked better questions even. But my mental model has been that, mitochondria is sort of an organelle inside the cell. And are there more than one? And so the number per cell is growing. That's what. Yep. Okay.
Also, mitochondria are bizarre in the sense that they are organelles, but they are not your organelles, meaning they have their own DNA. So they actually almost operate as a cell within a cell. So it's almost, it's sort of a parasitic symbiotic relationship.
Isn't there a theory that the mitochondria were a separate form and they got consumed by another one?
Yeah, that's the theory. That's the theory. It's interesting because you inherit all your mitochondria from your mother, right? It's a pure matrilineal line. It's not. There's no paternal transmission because they're intracellular, so they come from the egg, and sperm don't contribute. Sperm contributes DNA, but not mitochondria. So anyway, so yeah, the point is, in a training sense, the more you use a system, the bigger it grows. Your body always compensates for use by upregulation. So if you work a muscle, your body builds more structure, more protein, lays down more protein on that muscle to anticipate for further use.
So does this
chemically?
Is there a potential stressor that could, in a sense, push the body to fatten the thin blue line? And I'll use those semantics only because they're easy for video, et cetera, instead of repeating. Is there a stressor then potentially for doing, say, zone 3, 4, 5 training first and depleting to some degree the glucose pathway, and then doing zone 2 training where maybe that more available resource then is quite literally and figuratively exercised, right, more, or needs to be exercised more as a primary source of ATP eventually? Is that potentially a thing which has implications for the order in which you maybe would, construct workouts, where maybe you deplete yourself purposefully of glucose, do your whatever, and then go do your zone 2 training or something immediately thereafter to try to train up fattening that blue line?
It is an interesting question. It appears, as far as I can tell, that when this acetyl-CoA drops below a certain point, your body decides, which is right around, I think, zone 3, zone 4. Your body, zone 4 actually, more than zone 3 even. Your body decides that speed of reaction takes primacy and does not want to waste any generation of acetyl-CoA on fatty acid, and actually shuts down this enzymatic pathway. So
it's a maximum velocity curve.
Yeah, it becomes a maximum velocity issue. And so once the tank drops low enough, it actually shuts down contribution from fatty acid so that all of it is being produced at the maximum rate it can be produced.
And what's the recovery profile then? So suppose you did some zone 4 plus training.
Yep.
And you depleted, glycogen stores, et cetera. And then, I don't know, N number of minutes later, you go for that, slow jog or whatever it is to achieve zone 2.
Yep.
Are there known mechanisms? Is it temporally based or perhaps otherwise, energetically based where you re-engage that thin blue line and then therefore could maybe take advantage of that kind of a pattern in a workout to train up the fatty acid pathway?
So I don't know for sure, but what I do know is that what's interesting is when it drops low enough, it shuts off fatty acid production. But when you maximize this speed, actually other reactions in the pathway are slower. So this tank fills all the way back up again, but doesn't turn this on.
So that
So
that
there's some
does
other
allow
control
for the
mechanism that I'm not familiar with.
hmm.
Like I know it turns off when it gets low enough, but it fills
Mhm.
back up again because this is actually a really fast reaction. So it dumps it in really fast.
It's something in the just the higher
Yeah, but turning
acidity
the fat
conversion like that just that inhibits the whole process.
yeah, turning the fat burning back on takes quite a while, meaning it's not meaning minutes, meaning right, if not
Okay,
hours.
we got to clear all this crap out because we're producing more waste.
Yeah, I mean, so like again, you're burning all of this, right? This fills up, puts back pressure, pyruvate builds up, you create lactate. That's kind of the well, and again, there's argument that these should be switched, but functionally both, right? You end up with a pool of lactate. Well, as this starts depleting, even if the rate of burn is shut off, your body prefers to empty its lactate store and bring this pool back down to zero before it turns fatty acid back on, right? So it removes the byproducts of this pathway before it turns this pathway back on. Does that make sense?
Mm-hmm.
It's just,
And the
it's a homeostasis thing, right? Like ultimately, it's like
Well,
but
yeah, so lactate obviously when it's burned absorbs hydrogen, so it sort of decreases the toxicity to the cell. So there's advantages to getting rid of lactate preferentially over burning fatty acids, right? In terms of cellular health. That's my understanding, at least.
is the black line between the pyruvate and the lactose or lactase
Lactate.
excuse me.
Yeah.
Yeah. Super tiny on the screen. I'm trying to pinch to
that's
zoom,
right.
but that's not going to work.
No.
No, it's fine. I've been hearing it correctly, probably just whatever, trying to be precise and
Yep.
not sound as terrible as I'm now sounding. That said, that there's no directionality there. Is that intended bidirectionality, or is the continuation of that thick blackness coming into one or the other?
It's intended bidirectionality.
Okay, and that was where you were saying that there may be some debate about sort of the order in which those things might be put in a diagram. Okay, cool. So that, all right.
Functionally, lactate dehydrogenase is the enzyme LDH, and there's a ton of it in the cell. So conversion from lactate to pyruvate is exceedingly fast, or vice versa. In both directions, it's just an exceedingly fast reaction. And basically, the moment one is depleted, the other one will be converted to keep them in equilibrium.
Got it. And is the notion of the resources necessary for those things to convert a meaningful consideration, or is that like that can be sort of taken for granted in this macro view of the metabolic processes?
Before I answer, maybe I didn't quite understand the question.
I'm just thinking, you noted a reaction that is very fast that allows, I guess, the absence or preponderance of one to influence the other, pyruvate and lactate. And I'm wondering, is there any contributor enzyme, et cetera, that's an important consideration in the context of this diagram that catalyzes or otherwise regulates that reaction between the two?
No, not really. It's pretty much a free reaction. So those two are held in balance in a mutually interconvertible state pretty much freely.
Gotcha. Okay.
With the difference, so I will say this, and this is part of the argument actually that lactate should be the primary consideration here. Lactate can actually diffuse, this green line here shows it, can diffuse back into the bloodstream, whereas pyruvate cannot, right? It diffuses into the bloodstream, is taken up by other muscles and burned for fuel. So part of what's happening is if you're working your quads, your body has the ability to kind of burn lactate in your shoulder to give you more metabolic room, right? Instead of letting that hydrogen build up, right? It'll actually take that up and then absorb the hydrogen in another muscle from your bloodstream, again, to assist with the regulation so that you can keep working your quads for longer.
Okay, so the lactate, at least in intercellular, it's kind of just like a storage mechanism to like jettison out
a buffer.
local homeostasis and use homeostasis.
Yep.
Somehow.
It's actually, I mean, as a side note, it's actually kind of cool, right? Because like lactate also works as a hormone. So it also improves mood, you know, triggers other functions in cells distal to the cell that's
That are,
working. So
that are
anyway.
just based on energy, I guess.
Yep. So
Very cool.
anyway, yeah. So also one other fun little is there. You also actually have an uncoupler called what is it called? Thermoporin, I think it's called. Which basically in the electron transport chain just like opens this channel, right? And instead of producing any ATP, the hydrogens just like rush across it and it generates heat. And so it's also one of your body's cold control mechanisms. Is your body will just like, well, I don't need to actually do anything with the energy. I just need to release the energy. And so it'll actually just let hydrogen run down its electrochemical gradient and release or not release, it'll just become heat basically. So anyway, and I've noticed in tiny print here, let me zoom, I guess you guys can see it. When ATP is burned, about 60% of the available energy in the cleaving of that phosphate is actually used, and about 40% is lost to heat. So also part of how your body kind of heats up during activity.
Interesting.
I know you're not a veterinarian by any means. Is this energy chain understood to be consistent across life?
Yeah, pretty consistent. I mean, there are minor differences, right? Some enzymes in mice are a little different, but basically across eukaryotes, this is the system. Now, yeah, details in maybe like, okay, humans have 4 different versions of this enzyme, mice have 3, right? Tiny differences, but the core of this system is conserved all the way back.
Very cool. Okay, so then yeah, maybe is the time right to shift towards diagram semantics, and there are several dimensions to carve out here, and that's it, to some degree all the questions were inspired by just trying to pin down what the key dimensions were, and then we can ask this question, how do we sort of represent these things? And so we've got sort of high level with dotted vertical lines.
Sorry, there is one additional bit. I'm sorry. I should throw in. So I talked about what happens, this gets depleted. If the oxygen supply is insufficient, meaning your cardiovascular system, respiratory system cannot supply enough oxygen, right? This first part of the process does not require oxygen, right? So it's only once you get to the TCA cycle that you start needing oxygen as an acceptor for this electron transport chain. This part, the glucose part here, it only produces a tiny bit of ATP, but it does so without needing oxygen, right? And it does so very, very quickly. So it's a very fast reaction and non-oxygen dependent. So when
we say tiny bit, then we mean overall it produces a tiny bit for your body to use per reaction. Okay, so the fact that it's a faster reaction, there are maybe circumstances in which that could still be a meaningful total output of ATP.
No, it is.
Okay.
Here, let me show you this diagram here that kind of clears. So the rate of ATP production for creatine, for instance, right, is 73 millimoles per second, right? Doesn't make sense for ATP itself, right? But total available phosphates is 400, right? From glycogen up to the TCA cycle, right? Very, very fast, on the order of 40, but can only do 6,700 or so. From glycogen all the way through to the electron transport chain, again, takes longer, 16 millimoles per second, but you've got 84,000 phosphates. Liver glycogen we don't have to worry about. But from fat, you've got 4 million available phosphates, but the speed of that is only 6.7 millimoles per second. So absolutely, this glycogen to lactate is oxygen independent, right? Happens very fast, and it's a meaningful number, right? But it's not 4 million. Like it's not 200, which and to give you frame, actually, that's a great way to put it, is if you sprint all out from a standstill, right? You are limited by your ATP creatine phosphate system. Like that's before from the moment you start to the moment you have to slow down. The reason you have to slow down is you've run out of this 600 odd phosphates per muscle cell, right? So that gives you an idea. It's like 30 seconds to a minute worth of activity here. So then 10 minutes worth of activity at this speed is the total available of this non-oxygen dependent mechanism.
You can crawl forever. Yeah.
Yeah, I mean, and yeah, crawling is this 4 million, right?
That's
It's
what I
like
was referring to.
yeah, exactly. Yeah, exactly. I mean, you can go forever, right? Because it's like, okay, that's not doing anything.
My knees are sore, but I'm spunky. All right. Yeah. That makes sense.
I don't know that you want to use spunky in that context, actually.
Yeah, we
may
can
not
get there,
be.
I suppose. Yeah.
That may not have been the best
Yeah.
choice of words.
Yeah.
Yeah.
Very cool. Okay. Yeah. And the primary mechanism for, there is this, I mean, it's as expected, right? This is a very nonlinear system in that there are multiple variables, all of which are conflated essentially.
Yep.
Even in very obvious sort of mechanical ways, like, well, in order to take in more oxygen, you first do have to breathe out carbon dioxide. So you're just like, and I guess whatever, if you're falling out of an airplane, you could argue some gets in through your skin or whatever. But the idea is that there's mechanical and chemical and even at some level electrochemical sort of, conflations all across the board. And hence the difficulty not only representing the diagram, but also in comprehending and designing around interventions for such systems. So
the other challenge, I think, is just the fact that you've got cellular internal processes. We also, I think anybody to really understand it that doesn't have organic chem well up in their head, kind of the multicellular processes are probably a little easier to kind of grasp. If you can understand at a cursory level, at least, the metaphor inside the cell, this is all the same stuff. Because most of this metabolism stuff is that you were showing, it comes out of a cell and might get used by other cells, right? Like, so there's homeostasis inside the cell, there's communication, homeostasis outside of it, all baked into this system.
Yep.
Is that accurate?
Yeah, totally. And I mean, there's even neurologic inputs, right? So your brain, we don't know exactly how, but they call it the central governor, right? Which is why marathon runners stop rather than die, right? Your brain just literally won't let you. It will make you fall down and stop, right? Mm-hmm. There is some sort of central control that's like, yeah, no, you've depleted all the ATP, and if we let you deplete any more, you'll just die. And so we're gonna, even if you get eaten by the tiger, maybe at this point, the tiger killing you is a lower probability than you killing yourself. So we will make you fall down, right? And so, yeah.
So there's a monitoring and control system
at every level. Yeah,
alongside
at every
everything
level.
else, system and everything that's
a
switch and it's watching.
And compensatory. We talked about how increasing the thickness of the thin blue line from fatty acids, right? Similarly, it increases the number of capillaries you have, right, to increase the total oxygen supply, right? So your body adapts the cardiovascular system by building new blood vessels, increasing the size and strength of the ones you have in response to, hey, we didn't get enough oxygen and ended up having to do this non-oxygen dependent process, right? Or from the fatty acid system, like, hey, we're using this at maximum capacity. We need more. It's actually interesting because in terms of building blood vessels, zone 2 and zone 5 are the two systems that trigger new blood vessel production, right? Because in zone 2, you're kind of like using that oxygen system to its maximum capacity to some degree through the fatty acid system, whereas zone 5, of course, you're using everything to its maximum capacity.
Indeed. Okay.
Danny, the other thing I did want to say, so one of the other teaching conflations that happens is that, this system works differently if I walk at a constant pace, then I'm just using free fatty acid. When I do a VO2 max test, what I'm doing is I'm progressively opening this faucet wider and wider and wider every minute, right? In order to get a sample of what your system does and where the bottlenecks may be for you in this system, I sample progressively by just opening wider, wait a minute, open wider, wait a minute, and kind of seeing what your body does. But if I just wide open sprint from a start, my body doesn't go through that process, right? Like this gets depleted, this gets depleted, and instantly I'm going to be burning glucose because that's the fastest way to replenish it, right? So the rate of drop, very quickly turns on the right system without attempting other things, if that makes sense.
So this is related, to what we touched on earlier. Is it possible that there is an interesting test that may be somewhat related to the VO2 max, wherein one actually starts with zone 5 and then measures performance thereafter, where essentially the idea is let's deplete part of the system and shut off part of the system, and then ask the question, right? How are you performing in maybe the immediate term thereafter, or at various levels of rest and recovery thereafter, minutes, perhaps, if that makes sense, hopefully, or whatever. Then what's happening, with fatty acid, pathways, et cetera, that kind of thing.
I mean, you totally could. There's no reason you couldn't.
Process.
Yeah, there's no reason you couldn't. You could use the same equipment, right? If you were curious about, I've never seen it done, but the equipment would be the same. Is you just sprint, right, all out, till you have to stop, and then keep breathing into the device. It would measure because at the end of the day, the end products are consumption of oxygen. Oxygen is converted into water, right, and production of carbon dioxide, right. So as long as you're measuring the volumes of those two things, you're measuring this system in some sense, right? And the ratio between them gives you insight as to which pathway is happening.
Right. Yeah.
Right.
And the only thing that comes to mind, I wonder if there might be some intervention kind of thing that like I'll make something up here. Like maybe the target becomes something like, reducing the amount of time that your body needs to recover from zone 5 burnout. And can then and reactivates the thin blue line, basically, right? And maybe that measure might be indicative of overall system performance, and maybe to some degree the actual thickness of that blue line is the idea. Maybe there's some maybe it shakes out that
I've
there's
never
a
seen
reduction
it
there.
Yeah, I've never seen it done exactly that way, though. I don't know if that's just hasn't been done, right? The recovery metrics that are usually tracked, and they are important for performance, right? They are predictive of performance, is the rate of CO2 production decline is the primary thing. Is like, okay, you produced all this CO2 and your body's venting it off. How quickly does your body, when you stop running, how quickly does your body stop producing as much CO2, right? And so that kind of gives you a measure of like the efficiency of those recovery systems to downregulate and kind of process through the metabolites. So, so that is tracked but only for the first 2 minutes or so, right? So it's sort of like you should be able to reduce CO2 production by 50% in the first minute and a half or whatever. I don't know the exact metric, but something like that.
Yep. Well, and maybe just to mention it, and I think it was maybe obvious before, but just the thought there was with the vague assumption that the body will, as it does in other ways that we sort of know or whatever, self-regulate and adapt to the stressor or something. If we start kind of forcing the removal of one of the pathways, but then tell the body like, oh, but you have to keep doing stuff now. It's in
Sure.
zone 2, but you just have to keep doing it. Maybe there's some
Sure.
other secondary mechanism that says, well, we're switching on the thin blue line, and over time we're going to make it fatter because, like we ain't got nothing else, like, or that kind of a
Yeah,
thing.
so for that specifically, I don't know, and the reason is that, like, what I told you before is that when you're doing zone 5, the fatty acid burn is shut off. By contrast, when you're doing zone 2, if you haven't done zone 5 first, the carb burn is pretty much off, right? And so those two systems are already mutually exclusive. If you just do zone 2, it's the same as depleting your glucose and then doing zone 2 because the system was off already anyway. Your body wasn't even trying to use glucose in zone 2, right?
Gotcha. Interesting. Okay. So, all right. So this diagram as it is already like not terrible. It does have quite a bit of detail, and I presume that's where people might get a bit lost because you have to understand several dimensions here. The thicknesses, there's obviously a directional semantic. There's a layering semantic, literally with, kind of where you are outside moving into the cell, et cetera, that kind of thing. You've got oxygen input, output macroscopically. You've got the colors that indicate, up versus down regulation and enzymatic behavior, et cetera. There's lots of stuff going on here. I do think so. Just having gone through like this talk this way now, one thing that was useful at some level and sort of your presentation and the order in which you introduced things, I think was quite helpful. In seeing the picture where we originally saw sort of the cell wall, right? And then we saw the mitochondrial wall and the little sort of, barrier there, the two walls there with an intermediate that promotes some of this stuff or allows some of this stuff to happen.
Sure.
All that being said, I think that what was nice about that is mental model wise, one thing I took away structurally was that there were these like 3 primary pathways, right, for ATP production or whatever.
Yep.
And then there was lots of things going on, both microscopically to make that all happen. And then as we're seeing here, there's a bit of conflation between them, right?
Mm-hmm.
But those 3 primary pathways were kind of important and useful.
Yep.
And even just that basic mental model, I'm just thinking now as like, the most naive possible patient trying to understand,
I mean, that's the goal,
something
right?
about this. Yeah.
I mean,
yeah.
that's the
And
goal is so the issue I have is that this level of depth of explanation is not accessible to most people. Most people don't have the time, energy, patience, interest, or intellect to absorb all this, right? However, any more basic explanation I've come across is wrong and misses the point, and people then develop incorrect intuitions because it has lost key points. So the ideal is to figure out how to simplify the diagram. And I think already just with your delineation, you're right. There are 3 main pathways, right? There's free fatty acids, there's glucose all the way through the pathway, and then there's glucose without the oxygen component, right? And so, just realizing that and realizing those happen at different speeds, right? And then there's some interconnected control mechanisms that are driven by the speed of depletion, right?
Yep.
I think representing that would actually kind of get you a long way to getting people to have the right mental models.
Yeah, and then there's so what's occurring for me is there's the thing that I was seeing in my head, and it's actually derived almost directly from my diagram, was like there's these 3 sort of down pointing arrows, right? Those 3 primary pathways, and they all lead to ATP production. But then the other semantic that's important there, and there are various semantics that are important here, of course. I think the sort of biological layered semantic is a useful one for people to get a hold of at some level, maybe secondarily, but that's sort of like I think anytime you can imagine physically what is happening, as opposed to abstractly there are these reactions that are contributing to this energy source. If you can imagine and actually see spatially, physically what's happening, that does bring it to ground in a way that's useful in all science. That being said, the other thing that occurs to me here is also, I guess the patient probably is ultimately interested in and maybe is piqued then by what the sort of implications are, right? And so I'm imagining there are these three sort of, down pointing arrows. They have names attached to them, and that sounds great and is fancy and is educational at some level. There may even be some bars going across, two or maybe the outer two, certainly the center one, the two, that indicate at a high level the inherent conflation or correlation or shared resource pool, et cetera, that's going on here. And those don't even necessarily have to be named, but just this idea that these things are not completely independent, right? Even though we can identify sources. There's that. There's also the very mechanical input-output, the oxygen and carbon dioxide, and the implications that those things have. I can see those things being pumped in maybe to the left and to the right of this thing, if we think of that as the whole system, right? That's sort of a very external, internal, organism level kind of, atomicity going on there. That could be useful. And so something like that feels useful. But then the question becomes, right? It's one thing to nail what's actually happening, right? And maybe that's kind of close in some weak abstraction of everything, or maybe a useful abstraction. But then the question becomes, does the way we arrange those things, right, lend itself to the thing you were saying is often missed, which is what's the point? And do you get the right intuition for the thing that will improve your health outcomes? And then can you lean on that, right, clinically over time when things come up and you're like, well, look, we're doing this because you can see right here that this is the important pathway. Yes, it has this conflation, but we've addressed that for this reason. Now we're right here. And if we can do this, you get this outcome, right? There's this health benefit, et cetera. Or it's treating something or whatever. And so that's the part where I feel the fuzzy abstraction is like maybe kind of in V0.1, there's a version, a candidate version that could be coming clear there. But then there's okay, we need to make sure that this is always cast or projected onto the space of like health and outcomes. And that's where it feels slightly fuzzier. Maybe we can hit it from that dimension now.
Yeah,
and
that's great.
A bit.
Yeah. So, I mean, number one, and I'm going to backstep a little bit. I gave the example of a maximum sprint on the creatine phosphate system. That probably wasn't exactly correct. A better example would be like a close to max squat, right? Where like you're not even out of breath. Like if you do a 1 rep max squat, you're not even breathing hard, right? You just reached the limit of that muscle's contractile ability, which is not just energetic. It's also related to, it's also related to the protein strength and contractile ability and other factors. But energetically, it isn't producing a whole lot of carbon dioxide because it's not lasting long enough, right? So
it's kind of a 2 rep max in that sense.
Yeah,
you can move the weight, but you will fail quickly because of the amount of work you have to do.
Correct. That's maybe an additional sort of semantic there, right? So yeah. Anyway, whereas by the time you're doing 8 reps, you are breathing hard at the end of that set, right? Because you're having to vent off carbon dioxide. Carbon dioxide was being produced in sufficient amounts that your body feels the need to start venting that out, right? So that's in zone 4, zone 5 at that point, right? Anyway, what was I going to say? Outcomes, health outcomes. So VO2 max specifically, what it is the maximum rate of oxygen consumption your body's capable of. So at full sprint, right? And worked up to, right? So you work up to that full sprint, so everything's turned on. It's not just a 1 rep max squat, right? You got, your body gets a minute to kind of turn itself on, whatever. But at all out sprint, how much oxygen can you consume, which is a measure of your maximum metabolic rate. How all enzymes, all pathways, all things turned on, what are you capable of, right? For completely untrained individuals, usually there are limitations in the enzyme pathways, right? You don't have enough mitochondria, you don't have enough enzymes built up, et cetera, et cetera. For anybody that's reasonably trained, even, we're not talking about like Olympic athletes, but even somebody reasonably trained, the limitation usually is oxygen supply, right? So it's usually cardiovascular at that point,
Hmm.
right? And so, and to some degree, lactate burn as well, the ability to burn lactate. So, anyway, the point is, we know that VO2 max correlates exceedingly well with overall mortality and with cardiovascular mortality because that's sort of what you're testing. Very functionally, like, hey, what's your maximum ability to supply your muscles with oxygen, right? That's what your heart's supposed to do. How well can it do it, right, when really put to the test?
Or how well can the system facilitate that production is part of the,
correct. And that's regulated by a number of things, right? So like maybe you have asthma, maybe, but in general, your lungs' ability to absorb oxygen is rarely a limiting factor, unless you have a lung disease or something like that, right? Usually the restriction is cardiovascular supply, right? And so in terms of training, zone 2 with occasional zone 3 training is going to be the fastest pathway to increasing your VO2 max, right? There is some evidence that doing occasional zone 5 training just to hit your heart rate max, and by occasional once a week is kind of occasional, right? Whereas for zone 2, the stuff I'm reading lately is you need like 4 hours a week. And not broken up. That's the problem, is that like you really need it in chunks, like in an hour at a time, because otherwise you're not stressing that fatty acid system, right? You really got to be pushing that fatty acid system, and that just takes time. So you're talking about 40 minutes 4 times a week, or an hour 4 times a week or more, to really train up that zone 2 system, get all the cardiovascular benefits, get the mitochondrial benefits, et cetera, et cetera. And then for zone 3, that's kind of what they call sort of tempo training in running, is you would pick a pace that you can hold just barely for somewhere between 7 to 10 minutes, right? Like, and at the end of that 7 to 10 minutes, you're screaming, like you're burning, you're done, right? And that, in some way, my mental model, and whatever, maybe this is wrong, but my mental model is zone 2 is a little bit like hypertrophy training. You're just packing on muscle. And zone 3, zone 4 training is like strength training, where you start doing 2-rep maxes and strengthening the muscle you have, right? So you're not packing on more, but you're taking what you got and increasing the total tensile force it can produce.
I'm not as familiar with the details of the zone setup for exercise. This is all heart rate. That's the metric.
No, actually, so and that's interesting. You'll see descriptions of zones in terms of heart rate, which is intended to estimate this, right? So the delineation of zones should actually be, what do you call?
As measured as VO2 max.
Well, it should be relative to energy system. So zone 2 is defined from wherever your maximum fat burn is to the point where you're burning 50% carbs and 50% fats. In between so when your fat burn is maximum and when you're at that 50-50 point, energetically, that's zone 2, right? After 50-50, when your carb burn starts going up and your fat burn starts going down as a percentage of total energy produced, that's zone 3, right? Up until the lactate threshold. When you hit the lactate threshold, that's zone 4, and when you hit the creatine system, that's zone 5.
They've got they've got a few different breath devices that are like you breathe into to measure that ratio for the 5 zones. I guess the app shows it in 5 zones. It must be.
I'm not familiar, actually. Yeah.
What is it? Let me look down.
I know Lumen was one.
Okay.
Yeah, so the
Yeah.
problem with Lumen is that they advertise that they do this, but they actually don't have an oxygen sensor. They only have a carbon dioxide sensor. So they can't actually tell you the ratio. They guess from the total amount of carbon dioxide what your ratio might be. But obviously that's not, yeah.
So VO2 max.
It's a game show. It can be
Yeah.
fun.
That's right. Got a hair in my mouth from my beard somehow. Anyway,
This is
yeah,
all shade.
that's yeah. So no, I mean, I think that now Caliber Biometrics, which I've bought but I haven't tested for accuracy, has both. So that'll be interesting to see. I'm excited to break out that box and test it against the Pinoy to see how accurate it is. I'm a little bit trying to work out how to test it because I'm like, well, if I get on and run, then obviously that affects the next. If I switch out device, I don't know, right? I mean, I can test them lying flat, I guess, but which shouldn't change too much. But yeah, I'll have to work that out. What's that?
Day to day or something, like don't do it long.
Yeah.
You just get the incongruences.
Or
you
if I
could
don't go
probably
to zone
do-
5, like if I just go up from zone 1 to zone 3, rest for a good 30, 40 minutes, right? And then kind of do it again with the other device, see how well the curves replicate, and maybe repeat that back and forth twice so that each device has this is how it was affected by the previous rep or whatever, right?
Yep. That makes sense. That's what I'm suggesting. Even at the very least, you might be able to evaluate fairly reasonably well within like zone 2 or something. Because like, okay, you didn't burn out, you still had the resources or whatever, and like you can still kind of compare a little bit there. But yeah.
And one of the things I talked about actually too with that company is like, okay, maybe they're not up to reference grade standard like the Pinoy is. Like, okay, in the office we have a research grade calibrated XYZ, but if you're even in spitting distance, right, then the option of measuring your metabolism pretty directly on an 18-hour basis, it's kind of like, well, okay, maybe my heart rate monitor isn't EKG quality, but it's still used more. I can't carry an EKG around with me, right? So, but I can carry a chest strap, right, or a wrist strap.
That's perfectly good. Basic threshold.
Yep. So it doesn't have to be perfect, but if it's even in reasonable levels of accuracy, then the option to kind of monitor your metabolism for 18 hours a day at a time is actually kind of interesting.
Okay, I have a tactical thought or note here.
Yep.
How are we feeling on time? I've got some family just returned, which is why I muted myself for a moment there and also killed the camera so that I could communicate with someone peeking down into the basement saying like, hey, just FYI, we're back.
Yeah.
That being said, I do have some thoughts as we discussed cursory about, maybe some representations of this that might be appropriate and appealing to, those who are not seeking to dive any deeper kind of thing. And then mapping that stuff to the kinds of outcomes and interventions that would be appropriate as a function of understanding that stuff. And even it sounds like, VO2 max was one of the noted measurements that being able to put those numbers against this and in the context of this diagram could then be very interesting, especially from an intervention perspective, because then you can lean on that and say, okay, here's the data laid in some meaningful way against this concept. And now like there's sort of the deference to that ideal that you can lean on and say, well, clearly we need to work on this because the data says right here, like, that mental model gets sort of shaped.
Yeah, I think, it's interesting is that if you understand on some level that there are these pathways to generate ATP, what the VO2 max test is doing is serially testing each one of those pathways, kind of going one by one through those pathways and testing its efficiency, right? Then you can say, well, hey, in order to maximize your health, we take the least efficient pathway and work on that. If there isn't a least efficient pathway, then we do a combination of zone 2 and zone 5, and we kind of build up that base as far as it'll go so that the others are inefficient by comparison because you've maximized that base, and then we work our way up, right? And so, and realizing that zone 2, increasing that fat oxidation ability, is important for diabetes, for metabolic flexibility, and we don't have longevity data on that, but we suspect that that's a huge contributor as well, right? We do have data on VO2 max, right? That's a huge contributor as a predictor of mortality. And so these are the ways we can actually measure what matters, right? Is we can measure your
Yep.
metabolic health, your mitochondrial health, and your cardiovascular health in these ways.
What also occurred to me here was that at some level, and this is even related to sort of the loneliness discussion and other, psychological and otherwise well-being aspects to the practice, that, there's an opportunity for an intervention to actually address multiple dimensions of the well-being, for the patient. So for example, if it turns out this is one sort of bland linear example, but, if it turns out you need to work on zone 2 training, right? Well, great. Joining a run group is both social, works on zone 2, right? Has whatever, like there's it's interesting that even in the context of maybe a bigger diagram or an association of this diagram to other sort of the key dimensions that you've carved out, right? In the more holistic sort of realm of treatment, that there might be a lot of interplay there where,
I mean, for me, it would be a walking group or a sitting group that would be at zone 2, not a running group per se.
I think it would be slow build.
Yeah.
Give me an
and that's social. I mean, that's definitely social.
Give me some oxytocin.
Yeah.
There is a relation there.
Right.
No, but it's just thinking structurally about the whole thing, because it's there are always these sort of levels of perspective. And I know at the highest level, it's like, hey, we want to take care, but there are multiple dimensions, all of which are inevitably conflated. And then, now we can kind of maybe zoom in on a particular dimension of measurement, and then ask the question, right, from this measurement, how do we then intervene? And there is a sort of physiological component to that, and also maybe a psychological component to that. And that was maybe just one example of where those things converged a bit, where it's like, all right, well, you need to work on zone 2. So join a run group, not just to work on zone 2, but it's also social and healthy.
Sure.
And there's going to be a lot of that with like every
overlap.
For sure.
Which
Which is cool. And when you're leaning against, in the various levels, and hopefully not too many, or the levels themselves start to become an irreducible set of details, right? You want few levels so that those are the ones you can always keep in mind and they guide you. But like if there's this metabolic model, right, that's pretty important, it sounds like, in your overall health for various reasons. It's also measurable in a meaningful way via VO2 max, et cetera. You've got that model, you've got the big picture model that you've talked about, with the 8+ dimensions or so, that kind of a thing. Mapping and meaningfulness, there becomes a nice kind of convergence probably in some of the interventions where you could map out a plan for someone and be like, hey, well, look, the thing we're targeting for the next quarter or whatever it is, whatever the temporal frame is, we're actually addressing, like in this big picture, right? Like these 3 or 4 things, but we're doing it with like just a couple of interventions, which is pretty cool. Yeah. And you track those over time and you just see this like overall like filling of the of the pie kind of thing, and so you get to that place, like you said, where it's like, well, what do you want to do today? Ooch it forward in zone 4, like I don't know, like it's that's cool. Okay, that said though, what is the time like for you guys? I'm feeling a bit of pressure to break off and see some families in town from out of town today. But I am energized to think about further and refine some notion of graphic and drawing here.
Yeah, I mean, I've conveyed at least the model, I think, to you guys, unless, whatever. So, and I'm happy to share these diagrams or whatever, but, I mean, I think let's maybe just stew on this and kind of, I mean, even what you said, Jassem, in terms of like breaking down the 3 or maybe the 5, right? Maybe, say, okay, this is zone 1, 2, 3, 4, 5 in terms of like what the thing is of what's being burned in each of those zones. So start with 5 arrows just so that it correlates roughly, even if it's less accurate biochemically or whatever. I don't know. I think there's a couple ways to do that, representing again speed and volume, right? What's the speed of the reaction and what's the total pool that can be burned that way? And then, as you said, oxygen and carbon dioxide inputs and outputs, and Yeah, I mean, maybe
And
we
some
can just,
subtle measure of correlation across them that gives you that freedom or flexibility. Like when one thing happens in your mind, you're like, well, there's a trade-off here. It doesn't come as a
right.
surprise. It's like, well, you can see that connection here.
Yep.
It's weak, but sometimes, right, in various, special cases, it might actually be somewhat prominent. Like if you
Yep.
are that depleted, well, this thing becomes important now, right?
Yep.
So there's like that kind of dimension. Yeah, something like that. Yeah.
For instance, actually, so great example. In asthma, the issue is the inability or the reduced ability to vent off carbon dioxide, right?
mhm.
So then the
Oh, that's
acid, so then the acid level builds up. So then you can't do as much work because you get stopped by the production of phosphate and hydrogen. So
Makes sense. Yep. And that's even that's nice in the sense that with these primary, for lack of a better analogy, like pipelines defined, where they're all oriented in such a way that it's ultimately outcome motivated in a sense, right? It's ultimately the interventions that are derived from the picture are related not only to the picture but to the outcome that's desired. Then when people do have these special circumstances, for them, you can just paint across one of those, things like
Yep.
asthma, and it's like,
Yep.
this is important for you. Therefore, right,
Correct.
things start to fall out of that in the diagram very naturally.
Correct.
Yeah, that'll be quite nice. Yeah, I was going to make the request. You preempted it, which is awesome. Yeah, sending the diagrams would be great. I have seen them, but I can kind of digest them a bit further. And
Sure.
and yeah, maybe even next time, we can carve out some time to like draw some things that, despite being probably incredibly naive from me, might start to
Yeah.
anchor you.
No,
That I'm like, oh,
yeah.
what is the naive perspective on this? How does the model kind of, project from places of naivety, which is a patient's, place kind of thing.
I think that's the
Um,
ideal, right? Is having gone down to the depths with you guys, now the I don't know if I can even call your perspective naive anymore. Now you've sort of been walked through the whole thing, but at least it's not so intimately familiar to you that you end up without being able to simplify. You're like, okay, that didn't seem that important. Whereas I've kind of lost that perspective, I think. So
Totally. In fact, it's all we are in the position to do is to simplify. In the absence of deeper knowledge, we cannot go any farther. It's like, well,
that's the beauty of it. That's perfect.
yeah.
yeah, we can't answer any detailed questions, that's for sure.
Yeah. And you
Yeah,
can
that's cool.
remain content where like you have, in a sense, in a very vague sense, you have then sort of deceived us because we don't have all of the information,
That's right.
but maybe
That's
just enough that it might be useful to you.
That's correct. That's correct. It's a purely selfish semantic that yes, correct. That's
Yeah. This is awesome. And as a working
Cool.
session too, this is cool because it does serve as a nice instructional.
Yeah.
And then on the backside can be the sort of the synthesis, which is a nice little pairing. Cool.
Cool. Appreciate
Magic.
you guys.
Yeah, and I do
Yeah,
have
thanks for taking the time.
If you want to just do quick sketches, I can drive and we do it. I do have an app that I'm